Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Automating the Production of SDTM Datasets. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using the chat panel. And at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically for you. Please note that this event will be recorded and made available for future download. And at this point, I would like to thank Omnicom, who has helped develop the content for this presentation. Omnicom Systems is dedicated to helping the world's pharmaceutical, biotechnology, CROs, research, and medical device organizations maximize the value of their clinical research investments through the use of innovative and progressive technologies. Omnicom's electronic data capture and e-clinical technologies have been used in over 4,000 clinical trials around the globe. And now I would like to introduce our first speaker for today's event, and that is Albert Chow. Albert has over 20 years of experience in drug development across statistics, SAS programming, data management, and data standards development. He has worked in several major pharmaceutical companies and smaller biotechs. Currently heading up the data science group at SCRI Development Innovations, the CRO division of Sarah Cannon Research Institute. SCRI is a global strategic research organization conducting community-based clinical trials in oncology and cardiology through affiliations with a network of over 700 physicians in the US and the UK. The data science group is responsible for all data management, statistics, and programming activities for over 50 industry-sponsored trials and investigator-initiated trials in oncology each year. The group has been developing and implementing data standards spanning from CRF development through SDTM and Atom datasets to statistical outputs. Our second speaker for today is Keith Howells, SVP Development for Omnicom Systems. Keith has 20 years experience in the application of information technology to the drug development process. He's responsible for innovation for the Trial Master product line. Prior positions for Keith were as head of development for metadata solutions and as head of development for Oracle Clinical. And now, without further ado, I would like to hand the mic over to Albert Chow. Albert, you can begin your presentation when ready.
Okay, thank you, Andrew. Good afternoon, everyone in Europe, and good morning to everyone in the US. The first part of this talk, I'm going to talk about some of the challenges in SDTM mapping from the CRF data. And it doesn't matter whether you do it in some sort of mapping tools or do it in SAS, the challenges are very similar. So what I'm going to talk about is really from the CRF design and show you a few examples and how we map in the SDTM. Now, a lot of times people, when they map the data, they just think about the data fields and variables and how they map to each other. However, with some of the guidance that I'm going to give you here, it will make it a lot easier as well. So part of the talk I'm going to talk about is maybe following the C- standard and also give some consideration into control terminology and the compliance checks. And I'm going to show you a few examples of some of the challenges that's faced in some of the more complex trials, especially like in the oncology area. Recently, FDA has produced a draft guidance in providing electronic submission, providing regulatory submissions in electronic format, and specifically on standardizing the study data. Now, this is only a draft document at the moment, and so it's not binding. However, by reading this document, you'll know what's to come in the near future. Together with that, there's a conformance guide, which really specifies how the study data needs to look like in future as well. And one of the things I would like to call out is in the document itself, in the guidance document itself, it says that in future the data must be in a format that the agency can process, review, and archive. And what it's recommending is that to use the standard formats and terminologies specified in the data standards catalog. So one of the standards specified was the SDTM. One of the important points when it comes to designing CRF is to think not just on the CRF design part, but also thinking how you're going to use it further down the line, especially in SDTM, for example. One of the reasons is because you always need to map the data from CRF to SDTM. So ideally, if you start thinking about this process at the CRF design stage, it will really help you. And following the C- standard, that will really help when it comes to mapping in the SDTM as well. However, the C- to SDTM mapping is not prescriptive on how you collect the data. C- doesn't rule whether you collect the data in paper or ADC. And one of the important points is you need to make sure that you collect what's needed in the trial to answer the questions for your trial. Sometimes people said to me, why don't we just, we don't need to have this information in SDTM, or we can't map this data in SDTM, so we're not going to collect it on CRF. That's not true. You can always think about what you need to collect on the trial. However, just bear in mind what you need to map to afterwards. So what I'm going to go through is a very, very simple example, first of all, just to illustrate a few points. I'm going to show you a demography page, and I'll go through 3 of the questions: the date of birth, which is a date field, sex, and race, which are multiple choice questions. So first of all, with mapping dates, like date of birth, if you follow the C- standard, it will recommend that you collect 3 dates, possibly, the day, month, and the year. The alternative is to collect 1 date. So this is where you need to decide whether you're likely to have incomplete dates or you always collect the complete date. If you're always having complete dates, you may just have 1 field. However, if you have partial dates, or potentially in some countries where you're only allowed to collect a year, you may want to opt for this option of having 3 variables. But whichever way you do, if you follow the C- standard, you can always map it relatively easily into SDTM, and you can write a macro to be able to map all that information into 1 SDTM variable. And in SDTM variable, you always have the date represented in ISO 8601 format. So you've got the year part, the month, and the day. And if it's a date time variable, you will also have the hour, minute, and second. One of the questions you'll want to start thinking about when it comes to SDTM mapping is the length of the variable. So for example, here is the date of birth. If you're just collecting the date part, the maximum length you need is really 10, because it's just a year, month, and a day with the dashes in the middle. However, you need to collect the hour, month, minutes, and seconds as well. You need the length of 19. And apart from date of birth, you have a lot of other date time variables potentially. So you really need to decide for all the variables, do you have all the DTC variables have a length of 19? Or do you have certain rules where if you're just collecting date only, it will be a length of 10, while date time variables, you collect in the length of 19. The next example is the sex variable. Now, sex, for male and female, you'll have to refer to the control terminology, because in SDTM, for the sex variables, you always associate with the control terminology sex there. And this is managed by NCI. And one of the important part is you need to check whether the code list is extensible. In this case, it's no. So you can only use the submission values listed below, the F for female, M for male, U for unknown, and UN for undifferentiated. You cannot add any more codes to that. If you add more codes, you will be out of compliance with SDTM. There are other code lists which are extensible. In that case, you can add your own additional values in there as well. So you have to really bear in mind when you look at the control terminology which one you do. And you can find the control terminology from this website here. The next variable I want to talk about is the race variable. For this variable, sometimes in some companies you will collect the race as check 1 only, so you can only check 1 option. While in some other places you'll have the race where you can check all that apply. So how do we map the 2 different scenarios? So for the race, if it's just check 1 only, in your database you may have something like this, where you have the race and then another field for the other specified for the free text there. When you map to SDTM, you map into the domain DM, which is demography, and also you need to use the supplemental qualifier sub-DM as well. For patient 1, it's very, very straightforward because the race Asian just maps straight to Asian. For patient 2, although the race is other and the race other says white, really you need to map it to the DM domain as white and not other there, because this is part of the data cleaning you need to do. For the third patient, the other South African, what you do is for the race, you need to map the other, and then the race other, you need to make sure you put in supplemental qualifier, and the QVal will capture the South African value there. As I mentioned earlier, sometimes you may collect the race in a check order apply situation, so you can choose multiple options. In this case, how do we map that? So your database table, you may have one variable or one field for each option there. So when you map it, again for the first patient Asian, because there's only one box that's checked, that's relatively easy. You just map to the Asian. However, with second patients, because the site may have ticked white and Asian at the same time, so you have two choices there. So what you do in your DM domain, the race will be, the value of a race will be multiple. In the sub-DM, you put in the white and Asian in the QVal variable. Another example I would talk about is adverse events. The reason why I want to call this out is because more and more there are more treatments that are combination therapies, especially in oncology, HIV areas, etc., where each patient may receive multiple study drugs. And you want to capture the relationship to each drug and the action taken to each drug separately, because you may have different relationships and different action taken for each drug. So in this example, what I have is 2 drugs here, and you have 2 relationships and 2 action taken. So on the database, you have your 2 relationships. One may be yes and one may be a no to the second drug. And then the action taken, it may be the same or it may be different for the 2 different drugs there. So in this case, they're both the same. So how do we map this into SDTM easily? Because SDTM, you can only have 1 relationship and 1 action taken. So the way I recommend here is for the SDTM dataset, the AE domain, what you have is the relationship, you have multiple. So denote multiple drugs that you're looking at. And same for the action taken. And then in your supplemental qualifier, the sub-AE dataset, you will have the relationship for each drug separately. And you'll have the, for example here, the first 2 rows, AE rel 1 denotes the first drug, it's a yes. And AE rel 2 denotes the second drug, it's a no. Sometimes you may want to you may not want to actually use the exact control terminology on your CRF because you may want to collect more information. That is possible. You just need to make sure that your non-standard terms can be mapped to control terminology when you design the CRF. So, for example, here, you may have a study withdrawal page where you have more information on some of the reasons for withdrawal because we want to collect that information. This doesn't map exactly, this doesn't look exactly like the control terminology at this stage. However, what you can do is you can just, for each option, you can say what it may map to in SDTM. So in this example, the way you do it is in your DS domain, which is the disposition, your DS term will be what's pre-printed on the CRF. And then the DSD code, which has to be a control terminology, that would be what you use from the control terminology there. The next type of CRF I want to talk about relates to the findings. So this relates to laboratory data sets, the vital signs, ECG, etc. For findings like vital signs, some companies choose to collect the data in horizontal structure. So here you have height, weight, pulse rate, etc., etc. And each field becomes a variable itself. So typically, your data table may look like this. So you have height, a separate variable for weight, the unit, and then the pulse rate, etc. However, for SDTM, the findings domain, the data sets are represented in a vertical structure. So what you need to do is to transpose it from a horizontal structure into a vertical structure. And for example, like here, the pulse, you enter the information into the results and the status there. And for the unit for pulse, for example, you don't actually have that information collected on the CRF, so you wouldn't have that necessarily on the database. So you need to hard code that information in SDTM because you will need that unit in SDTM itself. Another possibility for the findings domain is to collect the data already in a vertical structure. So it starts to mimic what the SDTM data set would look like. So you may have your laboratory test, the results, and the units already mapped in a vertical structure when you in your database. However, this may pose some problems as well because, for example, like the collection date, the lab collection date, that is only for one record while you may have multiple tests on the same page. So your LB data, how do you put that into the data set? That's a question. And even if you manage to put that in, it usually appears on the first record, for example, like this. So the second and third records, even though it's from the same visit, you may not have the lab date populated in your original database. However, for SDTM, every single record, you have to have a date in there. So you'll have to somehow populate the date and make sure they are all in every single record there. With lab data, it causes a lot of challenges, actually, not just because of this horizontal versus vertical structure. One of the other things you need to make sure is there's a correct mapping of the LB test and LB test CD. One of the things that you need to make sure, first of all, is there's a one-to-one correspondence between the test and the test CD. So, and the reason why is because this is part of the SDTM compliance check. So if you have one test but multiple test CDs, you will get an error. Or vice versa, if you have one test CD but multiple tests, that will cause an error as well. And also be careful how you collect or which LB test or test code to use. So for example, neutrophils. If you're collecting absolute neutrophil counts, you may want to choose the test code MUT, M-U-U-T. However, you're collecting it as a differentials in percentage terms, you may want to collect it as a MUT LE, which is the neutrophils to lymphocytes ratio. This causes problem actually a lot in local labs because different labs may be collecting different units. Neutrophil is a classic example. So although you specify neutrophils, some of the labs may count it as absolute, while some of them may represent the neutrophil count as percentages. Then the next question is, because of the number of units that you can possibly have as well in different labs, so do you want to leave the unit as a free text, or are you going to use a drop-down list? And there are pros and cons in each one. Because you don't know exactly how many different units there are, so you may want to decide to choose the free text. By choosing the free text option, it's a lot more flexible. However, there's a variation of even how one unit is represented. So I'm showing you an example here is 10 to the 9 per liter. Even for that, different sites may be entering that 10 to the 9 per liter in 4 different ways. And when it comes to mapping from the original unit to the standardized unit, that can cause a challenge because you wouldn't know upfront how many different variations you've got. The other possibility is use a drop-down list. The challenge here is, will it be able to cover all the different possible units, even for one parameter? So here I have an example where it's just one parameter only, one lab parameter only, but there are over 10 different ways of representing units. And it may not, and the sites that you use may not even know which unit to pick exactly because what they see on the lab report may be slightly different from this one as well. On top of that, another challenge that you may have is a CRF page may map to multiple SDTM domains. So for example, like here on the vital signs page, although your database table, you may have just one database table called vital. However, the ECOG performance status, you will have to map it to the questionnaire domain DS. While the latter half of the CRF page, these are all vital signs, so you map it to a VS domain. To make it even more complicated, sometimes even one CRF field may map to multiple SDTM domain as well. So here we have an example showing the survival status page. So asking what was the subject status at this follow-up visit. If the patient died, you would collect the date of death and also primary cause of death. So on the database itself, you'll have all the information on one data table. However, when you go into SDTM, this information needs to go into 3 separate domains. So first of all, in the subject status domain, the SF domain, which is new in the new version, the SDTM 3.2, what you have is to have the original result saying dead, and the date is 2014-01-10. But because you collected the primary cause of death as well, so you need to have collected death details and put it into the domain DD. So here in the DD domain, you have the primary cause of death, septic shock in there, and also the death date in there. In the new version of SDTM, you have the DM domain. You also collect the date of death as well. So you put the date of death in the DM domain, and you have a flag that says to indicate that the patient has died. So you can see that even in this page, one field or one question, you can have multiple SDTM domains in there. Also, sometimes the data that you need for one SDTM domain may come from multiple pages. And this is best illustrated in the patient disposition dataset DS. So in the DS domain, you normally want to collect the information on informed consent, what happened to the patient at the end of screening, the randomization date, what happens to the patient at the end of the treatment phase, and maybe what happens at the end of some sort of survival follow-up information. So the informed consent obtained, you usually get that from your first page or in your screening visit where you have that informed consent date in there. The end of screening, whether the patient has completed screening and moved on to the next phase or whether it's a screen failure, that usually comes from an end of screening page. The randomization information, the date of randomization, may not even come from the CRF itself. So you may actually have that information from your IVR system or whatever randomization system they're using. And that could be external data that is not even available in the EDC system. The next part, the treatment phase, the end of treatment, you typically collect that information in the treatment discontinuation page. But as I mentioned earlier, more and more compounds are running in a combination therapy kind of scenario. So you may have treatment discontinuation page for each drug that you use. So if you have 3 or 4 different drugs, you have 3 or 4 different treatment discontinuation pages. And so you may need to gather that information from different pages and pull them all together. And lastly, for the survival follow-up page, typically that comes from an end of study page, or there may be some other pages you need to look at as well. So in summary, what I've talked about is when you build the ECRF and database, don't just build it without consideration of SDTM. Always make sure that you start having SDTM in mind as well, because you do need that information. Following the C- standard really helps with the mapping to SDTM. And make sure that you start looking at the control terminology and also attributes associated with data fields on the CRF, because all that will have an implication on SDTM and further downstream as well. CRF page may not be the same as SDTM domain. And what I've shown you is sometimes you have a one-to-one correspondence, but sometimes one CRF page may be mapping to multiple SDTM domains, or you can have multiple CRF pages information go into one SDTM domain, or you can have a many-to-many mapping as well. And that concludes my part of the talk. Thank you.
Great. Well, thank you so much, Albert. Before we hear from our next speaker, we do have 2 poll questions for the audience. And you can vote on these in real time by clicking on your screen. Your first question, what is your level of SDTM adoption? And your options on the screen are, we are not currently producing SDTM datasets. Your second option, we produce SDTM datasets only for studies nearing submission. Your third option, we produce SDTM for every study right from the start of the study. And your fourth and final option, we produce SDTM for every study when we're nearing the analysis stage of the study. Again, that question, you can vote on that in real time by clicking on your screen. What is your level of SDTM adoption? Looks like most of you have voted. I'm going to close polling and share the results of this first poll with you. And there we have it. Our first poll results, 48% of you said we produce SDTM for every study from the start of the study. That was followed by 27% of you saying we produce SDTM datasets only for studies nearing submission. 12% and 12% said we're not currently producing SDTM datasets, and the other 12% said SDTM for every study when we're nearing the analysis stage. Great. Well, we do have another poll question for you, and you'll be seeing that on your screen shortly. This question, in your organization, how much programming and QA effort does it typically take to produce SDTM data sets for a study? Is that less than or equal to 10 person days? Is that 11 to 20 person days, 20 through 40 person days, or greater than 40 person days? Again, you can vote on that in real time by clicking on your screen. In your organization, how much programming and QA effort does it typically take to produce SDTM data sets for a study? It looks as though most of you had voted at this point. I'm going to close polling now and share the results with you. There we have it. 42% said 20 through 40 person days. 25% said 11 through 20 person days. 22% of you said greater than 40 person days. And 11% said less than or equal to 10 person days. And with that, I will hand the presentation over to Keith Howells. Keith?
Great. Thanks very much, Andrew. If I can just start by commenting on those poll results, obviously the current level of adoption is pretty impressive. For the handful of you not doing SDTM yet, as Albert pointed out, the clock's now ticking. Now the draft guidance is out. There's a comment period, then it'll be final, and then there's 2 years after which you have to comply. So any submission after that time doesn't have SDTM, they'll just send it straight back to you. In terms of productivity, again, an interesting spread there. Obviously, those on the 40 and plus days, especially if that's towards the end of the study, that can go right on the critical path. Our own figures are in the 11 to 20 range, so some people seem to be doing very well indeed. But ours are towards the low end, and as you'll see, that is right towards the start of the study, so it doesn't actually end up on the critical path. Here's what I'll be covering, certainly starting with the approaches to SDTM generation, and then an overview of the specific approach we have in the TrialMaster product, and then really dovetail some of Albert's examples on specific challenges of how do you take the CRF data and map it to SDTM. We'll talk about codeless mapping, so if you didn't happen to start with the NCI standard code list, how can you do that? Some of these row transposition challenges where the way they want it in SDTM is other than the way you actually collect it on the CRF. Unions, there are a number of domains where you need to get data from multiple data sources. How do you do that? And then there are needs for aggregations, typically min and max, such as to calculate start and end of dosing, and then one slide on just customer experience to date. So there's really 2 approaches to doing this. Obviously, the assumption is the data is initially collected in EDC. All EDC systems produce raw data sets, typically 1 data set per form, and then you could take those and write programs in SAS or SQL to produce these SDTM domains. Take the 313 standard, for an example, there are about 35 domains there, averaging close to 30 variables each. So you've got kind of 1,000 variables to populate. It's really quite a daunting programming task. This alternative approach says, well, we'll do that right within the EDC tool. So we're going to do a mapping exercise between the data as we've got on the CRFs and where it wants to land in SDTM, just as an extended part of the study design. And if you can pull that off, a couple of things happen. One is you've got the expertise. It turns out to do this, you not only need programming skills, but you need medical skills to really understand where all these different pieces of data need to land. And those are typically the same skills that are putting the protocol together in the first place. And so doing it there, you've got that expertise while it's fresh, while the protocol is going together. And then obviously when you're actually running the study, the data doesn't have to move anywhere. So your SDTM is coming basically directly from the EDC database. So at any time you can say, please cut me some SDTM data sets. I want to do some preliminary analysis on the data. So this is a new capability. I'm going to talk through some of the basics of how that works. So first of all, the mapping exercise. So on the left, we have our typical EDC structure of visits. Visits contain forms, forms contain groups, groups contain items. sometimes it's just a one-to-one mapping. So if I've got an age item on my demographics, I can just drop that into the age variable in my demographics domain. Data type's right, the name is right, the units are right. Sometimes you might need a SQL expression, like the units aren't right, and so you've got to do some inline unit and measure conversion. Sometimes you have to concatenate variables together, so the unique subject ID is typically trial number hyphen site number hyphen subject number. So you've got to do a little bit of computation in doing that mapping. So that's the basic stuff, but sometimes you need to do something a little more complicated. So let's say you want to calculate the min and max dosing date. So because you're going to need that for computing the baseline flag, say in the vital signs. So the baseline flag is set to yes if it's the last reading before the subject started getting dosed. So what we need to do is first do a min-max and calculate those by patient. And then what we could do is take that source and use it to build some other domain. So now I can then take other things out of my raw data and join that into my computed domain and produce my actual SDTM domain. So those of you doing classical programming will recognize this as populating some temporary tables prior to actually doing the real work. And again, this can all be done in an iterative basis right within the single tool. So now let's drill into a couple of examples. So I've got a very similar example to what Albert used for demographics. So we've got a demography form here, and here's a piece of what we'll need in the SDTM data set on the right-hand side. So drop down for ethnicity, we might have stored it in our database as non-Hispanic, but the official CDIS code is not Hispanic or Latino, so we're going to have to do just a little inline mapping there. And you can see we've got to check all that apply race code here, and because more than one was checked, we're going to have to set that as multiple and then take those individual races and drop them into the supplemental domain, the SupDM domain. So how do we do that? Well, here's a piece of what the mapping screen looks like. We have variables and then their mapping. The variables are color-coded, so orange is required, yellow is expected, white is optional, so you know whether you've got to have a value for this or whether you can actually delete the variable or leave it mapped to empty string. And then on the right-hand side is what it's mapped to. So sometimes it's a one-to-one, so sex is mapped to gender. So implicitly, gender has got to have the CDIS code list behind it, MF, U, and UN. Sometimes it's just a straight text string, so we can just put that right in here. And then sometimes it's one of these little SQL expressions to do a calculation. So for instance, this ethnicity one is doing an inline lookup from the coded value in the EDC database to the standard terminology that SDTM requires. So again, this is just an inline lookup. So even if you didn't build your study according to C- standards, you can do this kind of inline computation to get to the required terminology. Now, the three race variables, those are supplemental variables, so they don't arrive by standard, but we go add them in because we need, where the race is multiple, to be able to say, well, the first race checked is this and the second race checked is that. And so we have a little function call here that says I'm going to pass in the values for each of the checkboxes, and I'm going to ask for element number 1 out of that, and race 2 will obviously have the same call, ask for element number 2. And that in turn calls a piece of SQL. So again, those of you familiar with database programming recognize this as a database function where you pass in the parameters and it passes back, in this case, a character variable. And all of this is done, again, within the study design tool. So there's a little edit window where you can create these functions and then call them. And because they're in the study metadata, they can be reused within the study. They can be reused across studies. Now let's move to the vital signs, where again you will typically do it in this portrait format. So I'm going to collect height as a variable and weight as a variable and the units for these as a variable. But this has to be unpivoted into a basically test code, test value structure, and then various others of units and standard value and standard units and so forth. So what we need to do is to rotate this data out to get to the structure that's needed, and then do things like unit and measure conversion. So we've collected it as 38C, but we want it to come out as 100.4 Fahrenheit. So how do we do that? Well, somewhat daunting on a first glance, but very elegant when you get used to it, is we have this unpivot operator, which basically takes these individual variables and unpivots them around, put them in a single column called, in this case, VS test code. So, and create a row for each. So in VS test code, we'll get one row for height, another row for weight, another row for body surface area. And what we want to do is, having unpivoted those, take the SAS name and drop that in the column. So as long as the study design, you've made the SAS name for these variables match the control terminology for VS test code, it will work fine. Ditto the SAS label for the full test description, and then the actual value for the result. Now, for the cases where you've actually got some inline computation to do, you can mix and match this unpivot with some of these little inline transpositions. So in this case, height units is collected as 1 or 2, so we basically say, well, if it's 1, then give me inches, otherwise give me centimeters. So this, again, is the control terminology for units. Ditto for weight units. All these other variables, this is the blood pressure, respiratory rate, and so forth. Those can be hard-coded. They're implied in the protocol. And then one more mapping for the temperature units. And then we can do something very similar for the result in standard units. So here's our familiar 9/5 of C plus 32 will give you Fahrenheit. So again, you're doing both inline transpositions and this unpivot all in one go to produce that vital signs domain. Moving on to dosing. This is an example protocol we tackled. We've blinded the drug names here, but did subject receive drug 1 and various information, the dose and start date and time and so forth. But then there's another one, because this is a complex oncology trial, and there's a slightly different one for drug 2 and drug 3, 4, 5, and 6. There's actually 6 different dosing forms in this protocol, but there's only one drug administration domain. So we have to take all these sets of data and glue them together into one domain. How do we do that? Well, in the mapping screen, as well as being able to map individual variables, you could say, please create me a union, very similar to a union in a SQL statement, where it says this set of data union, this set of data union, this set of data. So our first union is based on drug 1, and all of these references here, if I was in the tool, I'd be able to hover over them, and they would say, I'm coming from the first drug administration form. And then you just say, okay, give me another union, and then you map all those variables independently. So you see this is drug 2, and these would map to the items on the second drug administration form, and third, and fourth, and fifth, and sixth, and just keep going for as many actual dosing forms as you've got. And then you would typically use this filter icon here to filter out empty records. So, if only if there's actually a dose here do I include it. And now you get the complete set of all the dosing records across all the different dosing forms, no more, no less. And then what we need to do is, having done that union across our dosing forms, is to find the earliest and latest per patient, per subject. So we're going to need, in the demographics domain, for a unique subject ID, the reference start date. Here's this ISO date format Albert mentioned, which comes from this date concatenated with this time, and the end date, let's say, is this other administration form on this date and this time. So how do we do that? Well, first thing we do is create a reusable domain where we just have all the dosing dates. So we say, give me in this ISO date format the concatenation, the drug administration date, the drug start time. Then we create a second reusable domain that says, give me the min and max out of this guy, and they're both keyed on unique subject ID, so we'll basically end up with one row in this reusable domain per subject. And then we can just join that to the real demographics domain. So the mechanics of this is you build this thing, and then when you're building this, you type min and you drag this into here, and you type max and you drag the same thing into there. And then when you're building the demographics, this thing will be available as a source, and so you would just drag this guy into here and you drag this guy into here, and all of that is done. So again, very similar to doing temporary tables and joining to temporary tables, but the beauty is it's all done in the same tool in this very iterative manner. Finally, some of the other features that are necessary to pull this off. Again, I mentioned you can have a where clause per domain and or union to strip out empty records and not applicable records. The built-in ISO date functions for date and date times, they do partials, so if there is no month, you get the dash dash in the structure. All these domains have sequence generators. All of those are defaulted appropriately to the domain, so it might sequence per subject in the demographics, and it will sequence per subject and AE number in the AE domain. For supplemental variables like those additional race variables, those will automatically appear in the appropriate sup data sets. So those race variables, you would find there will be a race 1 and a race 2 variable for as many races were checked in the sup DM data set. So all of that comes automatically. You can have whole custom domains. And you can ask for automatic length calculations. So SDTM, rightly or wrongly, is built on SAS transport files as the physical delivery mechanism. That's quite an old standard. It's actually fixed character widths. So if you declare a variable as character 50 and you only put a 3-character value in there, then you've got 3 characters of data, 47 characters of dead space. And the #1 complaint from the FDA over FDA submissions to date is just too big because of all that dead space. And so we do just-in-time calculation of the column length. So at the point we're about to generate the data sets, we say what's the longest piece of data in this column and set that variable appropriately. So one day this column might be char 3, and then the next day it might be char 5 if a longer piece of data has arrived. And then in terms of delivery, it's a zip file. You get 1 SAS transport file per domain, an error domain if anything's wrong, and you can have that on demand and or scheduled. So having done all of this work, you could just say, you know, go run this job every night, so at any day you always have up-to-date data sets. In terms of real-world experience, we've done this for about 20 studies to date. Typically, the SDTM mappings are the third phase of the study design. First phase is your CRFs and visit structure. Second phase is your edit checks and dynamics. And then third is your SDTM mapping. So then they don't have to hold up go live if you're on a short cycle. But again, typically it's 2 to 3 weeks after the rest of the study design. First SDTM data sets are then produced as soon as you've got patient data. So the beauty is your statisticians can start analyzing these early data sets, checking they're structured properly, checking they pass all the validation, checking variables are landing where they belong. And if anything's wrong, that's just a standard mid-study change to go change some mappings or whatever it is. So the beauty is by the time the study is finished, all of that debugging, development, testing has been done, and you're producing your final data sets typically 1 to 2 days after study lock. In fact, we locked the study for a customer last Friday, and the last data change and the last data sets were both produced the same day. So in that case, that would be less than 1 day. In terms of what it buys customers, cost improvements, again, because you're just recapitalizing on the expertise that's already building your study, and you don't have to do things over again, like define all your lookup codes and your metadata and your data types and so forth. You're reusing stuff that's already there, so the cost goes down. Timeliness, again, is very, very good. And the quality, again, if you just outsource this to a classic programming shop, because you need medical knowledge as well as programming knowledge to do this, it typically burns a lot of QA time in saying, no, no, this isn't right, this goes here, this goes here. And it can just be a big hassle, where, again, if it's done by the same people with expertise in the protocol in the first place, then that quality rate is much higher. And again, that's what they're telling us. So in summary, this is a new capability. We've had the capability for about 3 years, but we had a new release last December, which added some of these final features like unions and aggregations and so forth. So we're really excited about just a brand new way of doing it. Your SDTM data sets are available while your study is ongoing. Anytime you can ask to see your data, and it really works. There's no trick here. We've production proven, and there's not a domain we can't do. So with that, let me hand that back to Andrew.
Great. Thank you to both of you for those very insightful presentations. Now, at this point, I would like to invite our audience to continue sending their questions and comments through right now using that questions window for the Q&A portion of the webinar. And gentlemen, I've received some questions already, so I'm going to start with the ones that we have here. Our first question, can this mapping tool be used with any EDC product, or does it come only with TrialMaster?
Yes, it's only with TrialMaster. So obviously we're reusing the metadata in the TrialMaster design and using the TrialMaster runtime engine for actually getting the data and reformatting. So I don't know if it's good news or bad news, but you couldn't use this capability with some other EDC system.
Right, okay. Thank you. Another question here. Is there any impact on system performance from producing SDTM within the EDC product?
Broadly, no. There is a substantial amount of work going on under the scenes to rotate the data out into a more accessible format to even start the process, and then you can see a fair amount of SQL that's going on. But that's all happening in a totally separate database. So the EDC data is replicated to a reporting server. It's then transposed into this reporting format, and then the SDTM operations work on that database. So there's never any impact on transactional performance because the hard work is going on a separate server.
Right. Okay, thank you. Another question here. What level of validation is required to ensure that the mapping has been performed correctly using this tool, especially for the more complex, less straightforward mapping?
Albert, would you like to start that one?
For me, I suppose, if we're just talking about general mapping, not using this tool itself, obviously you go through whatever the mapping, and people tend to double program and check it through. With the mapping, I think if you use the mapping, you still need to do certain level of checking. Obviously, the validation itself in the tool, that there is a certain level of validation in the tool to ensure that. But you still need to make sure that I would say something get a statistical programmer or statistician to be involved, making sure that the fields have been mapped to the correct variable for a start. I think that is important as well, because that's usually where some of the gray areas start to come in, with everyone having a slightly different interpretation. So you really want to at least have that level of input from the statistician, because ultimately they will need to use the SDTM for the ADAM and further down the line as well. Keith?
And let me add to that. So obviously there's no magic. The automatic stuff is to send the data sets through the open CDISC validator. So that will catch, hey, this required variable isn't mapped, or this value isn't in the code list, or the data type's wrong. So we do that automatically. And then the rest is QA processes that, again, just extended part of the study design, and then obviously the customer having a good look at that with their statisticians. In general, we're finding that the development effort is 5 or 6 times the QA effort. So generally, once you've put the investment into the development, the QA is relatively straightforward to confirm things have landed where they're expected.
Right. Okay. Thanks to both of you for that. Our next question, how much time does this add to the upfront database build?
Again, typically 2 to 3 weeks. It's still a lot of work. Again, you've got 1,000 variables to find a home for. It's not trivial. But you don't necessarily have to have all of that finished by first patient in. So if it's a phase 1 and you've just got very tight timelines, you might go live without the SDTM mappings at all, or with them only partially complete, and then carry on developing on the side and roll them out with the first mid-study change. But if you want a heuristic, it adds 2 to 3 weeks to the total effort, but that's never on the critical path to actually going live.
Right. Okay. Thank you. Another question here: Are you or is the system equipped to handle SDTM MIG version 3.2?
So we haven't productionized those new domains yet. The latest we have out of the box is 3.1.3. But it's all metadata driven. So we basically have database tables that say, here's the domains, here's the variables in them, here are the variable properties, and so forth. And then when you go into the mapping tool, magically there they all are. So we can, just with database scripts, roll out any new domains. So, when the cancer domains first came out, we rolled them in on top of 3.1.2, even though they hadn't, officially been put into the standard yet. Ditto for the device domains. So the good news is, as new domains come out, and they're coming out at quite a clip now, we can just write some SQL scripts to load up that relevant metadata, and magically there they are for mapping.
Okay, thank you. Our next question: briefly describe deriving SE and SV. How are trial design data sets set up? Sorry, I hope that made sense.
I can probably speak from a SAS programming angle. Obviously, the SE and SV, they're slightly easier to do in SAS itself. In terms of trial design data sets, there are multiple ways of doing it. I've seen people putting it into Excel and then reading into SAS. I've also know that some people actually put in EDC as a data table, and then that churns out to SAS itself as well. So I've seen multiple ways of doing it. Keith, do you have any comments on the SE and SV data set derivation?
So I'm struggling with acronyms. SV is subject visits and SC is subject characteristics?
Subject elements. So this relates to the trial.
This is study metadata, right?
These are per subject, each patient, what are the visits and what are the elements for each patient.
Okay, so we have the visit externalized as a CRF. So anything that's based on a subject visit, there's a CRF for that. And so, just querying that form gives you the distinct set of, or the discrete set of visits for that subject. So that's easy. But extending the question a little to some of the non-subject based information like trial arms and so forth, the trial definition form is extensible in TrialMaster, so you can go put extra pieces of information under the trial. So you could go put a repeating group of trial arms underneath there, and now you've got a source structure for building those non-subject specific domains.
Great. Well, thanks to both of you for all of those answers. We have reached the end of the question and answer portion of this webinar. If you do have any further questions, however, you can please direct them to the email addresses for both of our speakers showing on your screen right now. Thank you, everyone, for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event, and a survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our further webinars. Now, please join us in thanking our speakers, Albert Chow and Keith Howells. We hope that you found this conference informative today. Have a great day, everyone.